Hi everyone, it's Mr. Sinti, and today I'm going to talk to you about the urinary system, and in particular, I'm going to talk to you about urine formation. That's right, when blood comes out of our heart, it travels down the aorta, and one of the very first places it goes is into these two really important organs called the kidneys. And the kidneys filter the blood, they remove unwanted items such as water, excessive water, I should say, or excessive salts, and nitrogenous wastes such as urea, uric acid, that sort of thing. So let's get into this conversation because I think in the end, this is an important conversation because we can appreciate, all right, the kidney filters the blood. We can appreciate the urine when it's formed. It travels down these ureters and it's excreted, but I want to get into some detail in terms of like exactly how is urine formed. And in that conversation, what we want to talk about is the fact that when the blood is filtered, it's not filtered so efficiently to where things are easily removed. We have to reabsorb some of those things back into the blood. And so let's get into this conversation. So obviously, we need to review a little bit of kidney anatomy. And so we have this outer cortex, we have this inner region of the kidney called the medulla, and then this collecting area for urine, which is the renal pelvis. And as I mentioned before, the urine travels down the ureters. And blood enters the kidney via the renal artery, and it exits via the renal vein. That's deoxygenated blood. And of course, the kidney uses oxygen, but we really don't talk about that as much. We always talk about the exchange of fluids out of the blood that are being filtered. And all of that takes place in these functional units called nephrons. And so this is going to be a video discussing the physiology of the nephron. But I have to discuss a little bit of the anatomy. And so the nephron is partially in the cortex and partially in the medulla. It sort of dips down into the medulla. And all these tubes that are coming down deliver urine all the way into the minor calyces and then major calyces and then out. And so this is a conversation about the nephron. And so just a brief review of the anatomy or parts of the nephron. It starts off where blood is traveling ultimately from the heart, the aorta, renal artery, and then smaller and smaller arteries, interlobular arteries, until eventually it manifests into something called the afferent arteriole. And that blood is under very high pressure as it enters into the glomerulus, which is a ball of capillaries. The capillaries are where exchange of fluid occurs. So this is where filtration occurs in the glomerular capsule, sometimes known as the Bowman's capsule. But the renal tubule is different than the blood vessel. The renal tubule is shown here in yellow. The tubule, which completely surrounds the glomerulus, Bowman's capsule, then connects to this long tube called the proximal convoluted, because it's very windy, convoluted renal tubule. And so it's proximal because it's closest to the renal corpuscle, which comprises the capsule and the glomerulus. And so do you notice how it's convoluted? The convolution is really important because it allows for better reabsorption back into the bloodstream of things that the body doesn't want to get rid of, because filtration isn't such a clean process. And then it moves down into what's called the loop of Henle. This is the descending loop, and this is the ascending loop over here. And just point out how thin the loop of Henle is as it dips down into the medulla, which is down in this region right here. Then the renal tubule starts to get thick again, and it starts becoming convoluted, and it becomes what is known as the distal convoluted tubule. And then ultimately that connects to a collecting duct, and eventually the connecting duct will lead to the ureter and out of the kidney. Okay, so that's a brief coverage of the nephron anatomy. And again, here's a little close-up of that. So the blood's coming in, and when I say the blood's coming in, what I really want to say is that blood, as you may know, is composed of red blood cells. And the red blood cells do not diffuse out of the glomerulus. There's too large, they stay in. So the red cells, same with the white cells. The white cells come in and they come out. Red cells come in, come out. Large plasma proteins, such as what? Fibrinogen, antibodies, albumins, these kinds of plasma proteins simply come in and exit the efferent arteriole. And then the efferent arteriole becomes, this is a simplified drawing, becomes a capillary. And these capillaries surround the renal tubercle. And thus they're called peri, sort of like the perimeter around. Peritubular capillary network. It's a big mouth, it's a mouthful there. Peritubular capillary network. So all the fluid that is actually leaving the blood under tremendous high pressure, all that fluid is essentially plasma. Now, it's not plasma in a sense that it has the protein, but it has almost everything else that the plasma has. So the glomerular filtrate, mainly water, plasma, but also lots of salts, a lot of glucose, a lot of amino acids, that sort of thing. And lots of nitrogenous wastes, which are very small. But see, you don't want to lose glucose. You don't want to lose amino acids. You don't want to necessarily lose all of that water, which is coming through the tube. You want to reabsorb it. And so what the kidney does, what some people don't realize, the kidney filters the blood. It does. But it also reabsorbs the goodness back from the glomerular filtrate back into the bloodstream. And then anything that it doesn't want, it's able to secrete. And it's the detail, which is really cool, is this reabsorption, because it's really specific. And the kidney creates different tonic environments to facilitate the reabsorption of solutes back and forth between the renal tubule and blood. And so that's what this is kind of about. And so first off, a little bit, again, a close-up of the renal corpuscle. The renal corpuscle, again, is comprised of the capillaries and the Bowman's capsule. And again, you're just seeing a splice of it like this. It really goes all the way around. Here's the afferent, so the blood is coming in, the afferent coming in. And so if it's an afferent arterial blood has these endothelium cells, which are simple squamous, but it also is surrounded by smooth muscle cells. And I'm just going to put a shout out here. I won't mention it too much in this particular video, but there's another one that's going to be coming talking about an enzyme called renin. And these specialized smooth muscle cells of the afferent arteriole are called granular, because they contain little granules of enzyme called renin, juxtaglomerular cells. How's that? Juxtaglomerular, or JG cells. And that enzyme renin helps to control blood pressure. And so that's pretty awesome. So I won't get into that in a moment, but juxtaglomerular meaning sort of juxtaposed to where the glomerulus is, sort of opposite. So the blood's coming in, and it's under high pressure. It's under so much high pressure that if you're familiar with the anatomy of a capillary, it's very thin. The endothelium is made up of these simple squamous cells to maximize the rate of diffusion, but the pressure is so high that it would probably fracture those cells. And so they're actually surrounded by specialized cells, as you can see here with the nucleus in purple, sort of yellowish. They're sort of cells that reinforce the capillaries, the epithelium. They're called podocytes, or so they look like little feet. And so they protect those capillaries from breaking. But what I want to say about this is that the plasma comes in at such high pressure, it's just simply forced and squeezed right through the capillaries. And so when I say glomerular filtrate, that's what I'm talking about, is like this liquid that's exiting the blood. And I'm drawing it yellow in purpose because it's not going to be red anymore like the blood usually is, because, as I mentioned, the red blood cells travel in and out. white blood cells in and out, proteins in and out. It's really squeezing all this filtrate into the renal tubercle right there. And you're like, okay, so it's filtration, I got it. But there's some things in solutes in the filtrate that we would like to reabsorb. And so we want to reabsorb that back into the peritubular capillary network. So that's what this conversation is going to be about. And so let me just sort of emphasize this. And so this glomerular filtrate comes from the fact that you have this big Bowman's capsule like this. And then you have this afferent vessel coming in here, and then it branches into all these capillaries, right, that we just saw, called the glomerulus. And then, curiously, it exits also through an arterial that's a little thinner than this arterial. That's odd, because usually it's a venule. So that helps to even further maintain increased pressure in this glomerulus. And so what's happening is all of this liquid is leaving the glomerulus and entering into the renal tubercle. And that's referred to as glomerular filtrate or glomerular filtration. So all of this is entering in here, and you're like, well, what is it? What is it? Well, we can make a list. It's mainly water, again, and there's glucose. We have sugar from eating in the blood. We have amino acids. And then there's like a variety of potpourri of these little ions, principally sodium ions and chlorine ions and calcium ions and potassium ions and phosphates. And so all of this is entering into the renal tubercle. And I want to draw over here also urea. Okay, and that's that little tiny nitrogenous waste. In other words, like this. This is what urea looks like. It's a little tiny compound, and so it easily diffuses out of the capillary into the renal tubercle. And it's primarily this that the body wants to excrete. It's slightly toxic. And so, better drawing than the one I just made. But here's blood traveling in here. And here again, you can see the smooth muscles that make up the arterial of the afferent vessel are known as juxtaglomerular cells. You can see them right there. And I'm just going to foreshadow this a little bit, but they're granular and they contain the enzyme renin, which is going to be really important to secrete, which affects blood pressure. It's really cool. And so the blood, nevertheless, comes in. It's like zipping around. And this is where all the filtration, the glomerular filtration, occurs, and it goes into this lumen or space of the Bowman's capsule or glomerular capsule. All right, and so so many cool things. I'm like, the proximal renal tubercle. Notice here these cells. They seem to have like little extensions right here. They're epithelial cells, but as you can see, they're membrane right here, this apical membrane, not basal membrane, which is close to the bottom, but the apical membrane right here is made of the membrane is convoluted on a microscopic level. It's sometimes referred to as brush border. You might be familiar with this sort of strategy that the intestinal villi employs to maximize surface area when it wants to absorb food from the intestinal lumen. Likewise, you want the filtrate, which is all this juice, you want this filtrate to be able to be reabsorbed by these columnar cells, and then ultimately, let me add something. I'll just create this and make the efferent arterial become a capillary, a peritubular capillary. What we want is to get some of this filtrate to cross here and then enter back into the bloodstream. And so the thing about it is, if you know a little bit about biochemistry, you'll know that what I was listing before in the glomerular filtrate, such as sodium ions and chlorine ions and potassium and these things, all of those electrically charged molecules and glucose and amino acids, they do not just simply cross the phospholipid bilayer of cells. They must travel through membrane-bound proteins called facilitated diffusion to allow them to cross the cell membrane. So this is a good thing because that way the membrane proteins can control what we want to cross and how much of it do we want to cross. And I say that because the more transport proteins, the increased diffusion rate, and if there's no glucose transport proteins, then no glucose, or no sodium proteins, then no sodium will pass. So the kidney can control this, and it can also employ passive and active transport, and I want to get into that. Again, this is kind of detailed, but the coolness is in the detail. I hope you're, if you're watching this, I hope you're with me on that. So this is the most incredible photograph. It's a scanning electron micrograph of the glomerulus, and what we're looking at here is the blood vessels themselves and these specialized cells called podocytes. And these podocytes, like foot cells, have these like toe-like extensions, again trying to make sure that the vessels don't rupture under high pressure. And the filtrate literally can travel and it leaves the capillaries through these slits between the podocytes. And that's how it actually leaves the glomerulus into the Bowman's capsule, which is right there, I presume. So again, another picture. So what we're just looking at is the podocyte. See the nucleus here? There's the nucleus right there. Here it is here. And again, it's like sort of like toe-like. And so when the blood is coming in through the afferent arteriole, like this, it's under high pressure, and the filtrate is leaving through these slits right there. And it's going to enter into the proximal renal tubule, proximal convoluted renal tubule. And notice here that again the brush border and the columnar cells, and here are the podocytes, Bowman's capsule. This is a cross section of the distal convoluted tubule. These cells are a little bit colored. I won't get into it now, but I will in a separate video. This is the macula densa cells, which are really important because they can sense the concentration of salt in the distal convoluted tubule and therefore produce prostaglandins that will affect the juxtaglomerular cells, which are these cells that are lateral to the afferent arteriole, which will then produce renin and therefore increase blood pressure. So this, again, amazing picture is, see if you can guess what it is. It is the glomerulus without the Bowman's capsule. And so you can see the artery going in, arteriole going in, and here's the tuft of capillaries. So this right here is actually this. So this is drawing, obviously this is the real thing, scanning electron micrograph. And again, same here. There's multiple, there's again 1 million nephrons per kidney. So it's incredible. And so all of this is happening. You would need a microscope to see them. You could see them under the light microscope, but the scanning electrons just incredible. So you can see here there's a lot of filtration occurring. And so basically what we're talking about here is 3 main things that the nephron's doing. It's filtering the blood, which creates the filtrate. And then anything that's good, principally like we want to reabsorb water, we want to reabsorb glucose, all these things, that gets reabsorbed, and anything we do not want, we excrete into urine. And so these are the 3 mechanisms of the nephron. But again, if you look at this, you're like, wait a minute, this is kind of simple. The blood in and it comes like this. How come the nephron, this is a fair question, how come the nephron doesn't look like that? And it's like, well, it could. But I don't believe it would be as efficient, because the thing is, if you want to really maximize reabsorption, this is too quick. It's like beeline right out. It's like you really want to be convoluted, and you really want the capillaries to surround the convolutions. And so that's what I see when I look at the nephron. Not to be too silly about it or too corny, but I see like beautiful art and great design by natural selection. When you look at that nephron, it's totally an awesome structure related to function. And so it's just excellent. And so glomerular filtration, again, more of this. The blood's cruising in afferent. It's squeezing out into the Bowman's capsule or glomerular capsule, and it's going through these little slits that are created by the podocytes. These are the endothelium. These are the epithelium cells right there. Okay, and so fluid is filtering out into the proximal convoluted tubercle, and then the filtrate must then be reabsorbed into the bloodstream. Again, just to repeat, it's because some of the things in the filtrate we don't want to urinate out. You do not want to urinate glucose, for example. Okay, so it's all about reabsorption. And so reabsorption is something that I want to discuss. And so reabsorption, what I'm talking about is if this is the renal tubercle and the filtrate is coming in like this, the filtrate has some good things in it. And so the bloodstream is right here adjacent to it. Okay? And so when I say reabsorption, this is what I'm talking about. I'm talking about how some items in the filtrate need to be reabsorbed back into the peritubular capillary network. Okay? But as I've said before, sometimes, like for example, I'll just pick sodium, which is a positively charged ion. Sodium doesn't simply diffuse because it is positively charged. In order for sodium to pass through, you're going to need a sodium transport protein. That's right, a sodium transport protein that is specific for sodium, and then it can pass through. Does that make sense? I hope so. So what I'm talking about is there needs to be a permease or a transport protein for every specific filtrate ion there is. So there's one for chlorine, there's one for sodium, there's one for potassium, these sorts of things. Now, let me review some basic biology on this. Some of these ions need a transport protein to cross the cell membrane, but the reason they do cross the cell membrane is that molecules move passively from an area of high concentration to an area of low concentration. Now that's passive diffusion. No energy is needed. But there are occasions, and the kidney is one of them, where there are instances where the cells want to pump molecules from low concentration even into high, and that would require energy to do so because it's against the concentration gradient. And so let me just refresh you on that. Simple diffusion is often, we use like gases, like for example oxygen or carbon dioxide, and water can simply diffuse as well. Facilitated diffusion is what I was just mentioning. In other words, molecules moving from high concentration to low, passive, but yet needing a protein to do so, to pass through that phospholipid hydrophobic barrier, such things as potassium, for example. But there are occasions where the cell wants to push materials from low concentration to high, so that's against the concentration gradient, and that requires energy to do so, which is in the form of this particular molecule called adenosine triphosphate. The ATP, which is often produced by the mitochondria of cells, leaves its third phosphate. It causes the protein to change shape, and it can push the solute across, and then it becomes ADP. So it requires energy. And so let's look at this more closely. So as the filtrate is coming from the Bowman's capsule into the proximal renal tubercle, this is where most of the sodium ions are being reabsorbed. And so sodium ions are moving actively from the renal tubercle into the blood. Now, why do I say actively? It's because the concentration is higher in the blood. So you want to reabsorb the sodium in the proximal renal tubercle. And now what is the function of that? Well, in the proximal renal tubercle, one of the functions of that is if you can create a positive environment right here, then you're going to attract any of the anions, those are negative ions, such as chlorine or phosphates or bicarbonate ions. Any of these negative ions will come across sort of free because they're attracted to the positive sodium that's already there. Now, the bloodstream, again, in the proximal convoluted tubercle, is getting kind of hypertonic. Do you notice how there's a lot of ions or solutes in the area, which then will cause water to move across because water likes to go from high concentration to low, and what's lowering the water potential is the solutes in this area. So via osmosis, water is reabsorbed back into the bloodstream so that we don't lose too much of our water. Okay, so what I want to also say about that is, let's go here. So here is the renal tubercle. So the blood's coming in here, and it's like that. Okay, here's the glomerulus, and here's the efferent. So blood in, blood out, like that. Okay, and so the filtrate's coming in. Okay, so here's the filtrate, like that, like this. It's cruising in, cruising in, cruising in, and its filtrate's coming in like this. Okay, so let me emphasize something that's happening. Do you see, like, this is referred to as the loop of Henle right here? I've been talking about the proximal convoluted tubercle. I want to talk a little bit about the loop of Henle. Now, I draw this line right here because everything above this line is the renal cortex, and everything down here is the renal medulla. Now, one of the very first things that I want to discuss is that in the ascending loop of Henle, there's a lot of membrane proteins for sodium. That's right. And these membrane proteins, transport proteins, can actively, with energy, pump sodium from the renal tubercle into the medullary area. And so this area becomes really high in salt. Okay, so it's becoming really salty down in here. So it's using energy to do so. Now, what's curious over here is that there's no sodium ion transport proteins over on this sort of thin area. And so as it turns out, when water, let's go blue, when water is traveling down in here, and it starts to get a sense that it's very, very salty in this area, water will then exit the renal tubercle. Okay, and then where is that going to go? That's going to be picked up by the blood ultimately, because I don't want to make this too messy, but the blood is circulating around like this, and so that's going to be picked up by the bloodstream. And so what's really cool is that by the loop of Henle tries to create a very salty environment, and in so doing, it uses that to be able to reabsorb water. And so I find that that's particularly interesting. Now, if you look at the details of this, let me come back over here. Like, do you see over here in the proximal convoluted renal tubercle? I could say that when the filtrate comes in, I mentioned this before, when the filtrate comes in, one of the things in the filtrate that you do not want to urinate out is glucose. And you're like, all right, so maybe I shall reabsorb it. And that's what happens. It is reabsorbed into the bloodstream. Glucose goes into the bloodstream at the proximal convoluted renal tubercle. And you're like, well, how does that work? Well, it's kind of cool. Like, this is a close-up of, again, the convoluted proximal renal tubercle. This is the sort of the basolateral. This is the base of it. Here's the capillary. This is the apical brush border. So this is the filtrate right here. So this is the stuff coming out of your glomerulus, and then here's glucose. Okay, so let me draw your attention over here for a second. In the proximal convoluted renal tubercle, the cell actively pumps out sodium. And now, if you recall the details of this, it's sometimes referred to the sodium-potassium pump. the cell expends energy in the form of ATP, and it literally pumps out 3 sodium ions and then 2 potassium ions come in, and there's an exchange going on. So as a result of pumping the sodium out like that, it's making the sodium inside the cells of the renal tubule low in concentration. And it's also making it slightly negative on the inside. And so what that's going to do is it's going to therefore attract more sodium back out of the filtrate into the tubule and then back into the blood. So it actively pumps it out over here to draw more in. And you're like, well, that's a little weird. Well, one of the reasons why it draws it in is because there's this co-transporter that, well, when sodium goes in, it's sort of like those revolving doors when you go into it, like a big hotel or a big office building. They swing around, and like two of you can get into one rotation and both go in at the same time. This is a way in which the cells utilize the sodium movement to couple the transport of glucose into the cells from the filtrate, and then glucose then travels into the blood. So it's a detailed look that it uses active transport to then allow glucose to come into the cell along with sodium. So this is where the energy is being put in over here, but it has great benefit. So I find that detail kind of interesting. So all of that is happening right here. So, all that detail was basically what we're saying is the cells are reabsorbing glucose, they're reabsorbing sodium at this point. And again, this is the medulla, and it's very salty down here. It's very salty in this area because we're actively pumping, so a lot of ATP, actively making it very, very, very salty down here in the medulla, so that we could allow the osmosis of water. So water is leaving at this point. Okay, pretty interesting, huh? And then up here in the distal, we could still have some diffusion of other things, like for example, we can have diffusion of calcium. And I'm not mentioning everything, where like we can even have the active transport of chlorine and potassium as well. So sodium reabsorption and water reabsorption are linked together if you've been following this. And so as the sodium ions are reabsorbed through active transport, that allows for the passive movement of water out of the loop of Henle through osmosis. And so as you can see here, and again, a really nice diagram, water is being reabsorbed. In other words, we don't want to urinate out too much water. And what's fascinating about this, let me bring this to your attention, that this area right here is very thin, and it allows for osmosis to occur. So reabsorption of water, reabsorption of water, reabsorption of water. And then this thicker area, the distal convoluted tubule, doesn't allow for much osmosis to occur. So no water is leaving. Just a little bit of water is leaving when it gets into this salty area, but not a lot. But then what's interesting is if the body senses the fact that it's running low on water, in other words, it's becoming dehydrated. If it's becoming dehydrated, the body can actively try to attempt to reabsorb water, more water, and not urinate a lot of water. And so it's really cool how it does that. It does that in the collecting duct. Okay, and so that's what I want to talk about. So most of the sodium ions are reabsorbed before urine is excreted, as we mentioned. Now normally, the distal convoluted tubule and the collecting ducts are impermeable to water unless a hormone called antidiuretic hormone is present. Now antidiuretic hormone, if you recall, is produced by the pituitary gland, which is this gland at the base of the brain, right below the hypothalamus. And there's an anterior lobe and a posterior lobe. It's the posterior lobe that is producing this antidiuretic hormone, and the target is the collecting duct of the nephron. And so what's awesome about this is that the antidiuretic hormone will bind to a hormone receptor in the collecting duct cell, and then that will cause a secondary signal message, because this is a peptide hormone. So you get these secondary messengers, principally cyclic AMP. What will happen is it'll get some genes to be transcribed and then translated into proteins. Those proteins are shown here in purple, and then they're put into vesicles when they're removed from the Golgi apparatus, and then these fuse with the plasma membrane. And you're like, well, what are these? These are special proteins that will facilitate a massive amount of movement of water. That's right. These specialized proteins are called aquaporons. And these aquaporons, we used to think that just water moved passively in and out of the cell membrane, and it does. But when you have specialized transport proteins for water, then you can really get a massive amount of water to move all at once. And so the collecting ducts actually have the ability to produce aquaporons in the presence of antidiuretic hormone. So antidiuretic hormone can help water reabsorption, because normally you would urinate out the water, but if you're feeling dehydrated, it helps to prevent further dehydration if you're then reabsorbing more water. And then again, the brain controls thirst, which then allows you to consciously drink more water, which will then obviously prevent you from being dehydrated as well. And so just a refresher that urine is mostly water. It does have some solutes in it, but it's mostly urea is what we're trying to urinate out. And so let's talk about the whole urination process. And so when urine is eliminated, that ultimately the nephrons are producing the urine, and when it's coming down those collecting ducts, they go into the calyces, and then the renal pelvis, and then ultimately into the ureters, into the urinary bladder, and then maybe into this bathroom stall right here. And so here are the kidneys releasing the urine down the ureters here, as you can see. These are blood vessels. This is the aorta, this is the renal artery, this is the renal veins here, this is the inferior vena cava. These are carotid arteries here. And then these are the ureters coming into this urinary bladder, and then the bladder fills up with urine, and then it's excreted to the outside via the urethra. Okay, so if you wanted to look at the inside of the ureter, it's in other words like cross-section like that of the ureter, this is what it looks like. It's composed of 3 layers. It has this mucosal layer here, and this is where the urine is in this lumen. It has this muscle layer, smooth muscle of course, and then this fibrous coat on the outside. Here's a nice x-ray of this. You can see here's the renal pelvis where the urine is being collected, and it's going down the ureters. This is sometimes used to for physicians like to diagnose if there's some salt crystals, sometimes known as kidney stones, that will form in the renal pelvis and get caught in these minor and major calyces. And sometimes a kidney stone will get caught in a ureter. And so sometimes an x-ray will expose that. And then here's a nice shot of the bladder here. And so ultimately, the reason that urine is traveling down is obviously gravity, but there's also smooth muscle wave-like contractions called peristalsis, which helps to move the urine down into the urinary bladder. Urinary bladder is really fascinating too if you looked at its structure. So it has this muscular surrounding, and the ureters actually open up in the sort of the floor of the bladder, and there's the 2 openings down below where the ureters come down, and then the urinary bladder opens up into the urethra. Now this is male. How do I know that? Because the urethra is then surrounded by this reproductive gland called the prostate that produces fluids, mainly alkaline fluids, which then associate with the semen in order to keep the semen alkaline, so because it's entering into the birth canal. And so the urinary bladder is made up of four coats, and this inner layer called the mucosa is pretty interesting. It's this special type of epithelial tissue called transitional, and it's at times when the bladder is empty, stratified, and when the bladder is really full to almost maximum, it looks simple. In other words, it's transitional, it can sort of stretch out. And then you have this submucosal layer, and then again the muscle layer. Detrusor muscle is if you're pronouncing that, detrusor muscle is helpful in allowing the bladder to accommodate the urine that's inside. And again, the very outside of the bladder is the serous coat. Detrusor muscle helps to surround the bladder, and together that muscle combines with this internal sphincter muscle. Now, this is a really cool micrograph showing the mucosal layer of the transitional epithelium, and this is the submucosa here. So the urine would be over here in the bladder lumen right there. And so here's a cross section here of the bladder. You can see inside. Here's the ureter connecting to it. This is what I mentioned in terms of the urethra traveling through the prostate gland. And you're like, well, I mentioned semen before. Now that's entering through a different duct. Do you see this duct right here? This duct is called the vas deferens. This is taking semen, which is produced in the testicles, up from outside of the body into the body, around the urinary bladder. The seminal vesicle fluids are added here, and then that travels also through the prostate gland. Now, this muscle right here is really important. That's the sphincter that's helping to regulate urine flow out the urethra, which is, in a male, inside the penis. This is the erectile tissue that can fill up with blood, causing an erection. But normally, the penis is flaccid, and so urine can just travel simply from the bladder out the urethra right out this way. Now, in a female, this is the rectum here and the anus. This is the uterus. This opening to the uterus is the cervix. Here's the birth canal or vagina. This connects to the fallopian tubes right here. It's a fimbriae ovary. But here is her urinary bladder, and it's protected by this bone called the pubis bone right here. But what's interesting is you can see here there's a catheter being put in here to drain the urine out of the bladder. That's what this thing is right here. This cord is not normally present. The urethra is a little shorter in a female, and so the female has three openings over here. It has the urethra opening, it has the birth canal opening, or the orifice of the birth canal, and here's the anus right here. So again, here's now a shot of the male. This is the prostate gland here, and the openings right here of the urethra are sort of in this sort of triangular base called the trigon right in here, and the detrusor muscle, I have a hard time saying that, is up here, and that sort of combines with this internal urethral sphincter. Now, that is important along with the external urethral sphincter. This external one we have conscious control over, which allows us to be able to, pee or not pee. And so this becomes a problem in certain individuals, and I'll just bring up one example. Like, for example, if there's a problem in a male and it's detected prostate cancer, and maybe the physician wants to prescribes the removal of the prostate, sometimes this external urethral sphincter is removed, and you have no control over your urine output, and so you literally have to wear protection like a diaper to be able to collect that until you begin to the muscles begin to reform, and then you become capable of being able to control it or maybe not control it. This is the posterior view of that, and you can see here is the the seminal vesicles. These are the vas deferens here, and here's the prostate, and then urethra. So the act of urination is micturition. So micturition is, in other words, urinating. And so as urine leaves the bladder, it's by this reflex. And so the reflex is a combination of the bladder contracting along with the external urethral sphincter relaxing, which then allows the urine to be able to leave the urinary bladder. This is where it's contained. And so sometimes the mere stretching of the bladder, again, the transitional epithelium, triggers a reflex in the spinal cord, which will then allow nerve impulses to cause the muscles to contract and relax, if you're talking about the sphincter, which will allow urine to be excreted to the outside. So it's this external urethral sphincter that is controlled consciously, and that's located again right over here at the base of the prostate gland. Okay, and the urethra, again, for the female, is a little bit shorter, and here's the sphincter that controls urination in the female right there. And then in the male, it's a little bit higher up here, close to the prostate gland. All right, and then urine is released to the outside. And I just want to finish this whole conversation with the fact that if a female's watching this and you're like, what I'm really interested in is if I become pregnant someday, what's the story with needing to urinate, every half an hour to an hour? And it's especially late in the third term of pregnancy when the fetus is really huge like this. This woman's about to give birth any minute. You can see here the urinary bladder is quite compressed. And so this is what we're talking about. This is the urinary bladder and urethra right there. So this is why there's a tendency to need to urinate. So I hope this was helpful, talking about the function of the kidneys, one of the greatest structures of all in the body, the nephron. And so I hope you're interested in that enzyme renin. I'm going to make another video just on that and how blood pressure is controlled. So thanks for watching.